Hello lovelies, in this video we're going to be looking at light dependent reactions for your A-level biology. Now there are lots of new words in this, there are lots of diagrams to sketch in this, so make sure you've got your array of different colour pens and pencils ready so that we can highlight the important bits and put it in our folder to refer to later.
Hi everyone. Okay, so today we're going to look at photosynthesis proper, so the actual biochemistry of photosynthesis. We're going to start with the light-dependent reactions. So we've mentioned this already before, but the light-independent reaction is one part, and that takes place in the stroma of the chloroplast. We're going to look at today the light-dependent reactions, which take place in the thylakoid membrane because they need those photosystems that we talked about in the last video. So we're going to use light that we're absorbing, light energy that we're absorbing, and this is the first part of photosynthesis. Okay, so the first part we're going to talk about is something called photoionization. So anything where we've got the word photo and ionize, photo, it means light, don't forget. When light hits our photosystems, and you can see that here, so my light is hitting my photosystem, and as we talked about last time, it's going to go, the energy is going to go through those accessory pigments, it's going to be absorbed and then transfer through the accessory pigments to the chlorophyll molecule at the bottom. So when light energy hits it, and the light energy is transferred to the chlorophyll molecule, and that's used to excite an electron. Sometimes we talk about the fact that it's actually exciting 2 electrons at a time. We're exciting electrons, and what we mean by exciting is they're going to give this electron or these electrons energy. So we're transferring the energy and we're exciting the electrons by giving them energy. And what we mean by excited is we're transferring them then to a higher energy level. So you'll often see an arrow going up. This means that they actually have enough energy to leave the chlorophyll molecule. So this arrow going up is representing them going to a higher energy level and they're leaving the chlorophyll. And they're going to be accepted by a protein which is known as an electron acceptor or an electron carrier. Because chlorophyll has lost at least 1 electron, it has now become an ion. So an ion is an atom that has a positive or negative charge because it's lost or gained electrons. In this case, we've lost electrons. So my chlorophyll has become an ion with a positive charge. So because light energy was used to excite this electron and give it enough energy to leave the chlorophyll molecule, this is the process is known as photoionization. We've ionized chlorophyll, we've turned it into an ion by removing electrons, and we did that using light energy to excite them to let them leave. So this happens at both photosystems. So light energy is absorbed, and that light energy is passed to electrons, and that raises the electron's energy level, and they leave the chlorophyll and they go to an electron acceptor or an electron carrier. That's another protein in the membrane. So this happens at both photosystems in the same way. So what happens to that electron once the electron has left chlorophyll? Well, as we said, it binds to a primary electron acceptor, and then it moves down what we call the electron transport chain. So it moves along various proteins that are embedded, various proteins which are electron acceptors or carriers that are embedded in the thylakoid membrane. So it will go through or along a couple of these. So when this is pictured, so you can see here my electron comes up to my primary electron acceptor and it's moving down this electron transport chain to photosystem 1. So from photosystem 2 down to photosystem 1. And this is often a diagonal line down because what's happening as we go down the electron transport chain as an electron, but every time we pass through to another protein, we're actually losing energy. Each protein in the electron transport chain, each protein carrier or electron carrier or acceptor that we're moving in onto has a lower energy level. So each time the electron is losing energy. How do they move between the proteins? So they pass through a series of redox reactions. Re meaning, or red meaning reduction, and ox meaning oxidation. So if you remember from chemistry, oil rig so oxidation is loss and reduction is gain, then you can kind of remember how to do it. So oxidation is loss of electrons or of protons. Reduction is gain of electrons or protons. So they alternate. So proton carrier, so say this proton carrier here would get an electron. If it gains an electron, it has been reduced. And then the electron leaves and passes on to the next one. The electron's been lost. And so that protein in the chain has now been oxidized. This one has been reduced. Then this one loses that electron again. It passes it to the next one. So therefore, it's now been oxidized and the next protein has been reduced and so on. So we say they alternate redox, reduction, oxidation of each protein in the electron transport chain as they move along. Now, why this is important is because as they move along that electron transport chain, as they lower their energy levels repeatedly and they alternate between their redox reactions and they lose that energy that's being lost is then used to generate ATP. And we're going to talk about how we do that in a couple of slides' time. For now, we just need to say it creates ATP. So the energy lost through the electrons moving down this transport chain generates ATP. And in this first part of our sequence of the light-dependent reactions, the first electron transport chain we encounter is where the electrons have left photosystem 2 and are traveling down to photosystem 1. When these electrons are then passed to photosystem 1, you'll notice again my light energy coming in so they can be re-excited, energized, ready to go down another electron transport chain. So it happens in the same way in both photosystem 2 and photosystem 1. So talking about oxygen usage, when there's high light intensity, so when there's really high light levels or during the middle of the day, when the most light is being absorbed by the chlorophyll, there's going to be a high rate of photosynthesis. And when this is very high, it's often higher than the rate of respiration. So more photosynthesis is happening than respiration is happening. So the oxygen that's being produced from photosynthesis, it's actually, there's loads, and we don't need as much. So some of it can be used for respiration, but there'll still be some left over. So there'll be still oxygen in excess. And so that's when it most likely it will diffuse out of the stomata and out of the leaf. And that's really important because that happens every day during the highest temperature points of the day or when there's high light levels. There is often a lot of excess of oxygen, which means it diffuses out of the leaf. And that's how we have oxygen in our atmosphere. And it's essential for us to breathe. It's how we have an amount of oxygen in the atmosphere that supports all of the life on Earth. Okay, so now we're going to talk about how we actually make ATP through photosynthesis. Some examples we want you to talk about this here and talk about it in the context of photosynthesis. We're also going to come and touch on this in a very similar way when we do respiration. So we're going to talk about it in terms of chemiosmosis, or chemiosmotic theory, which is how we're making ATP using this electron transport chain that we've been talking about and the energy that's being lost from those electrons. As the electrons pass down the electron transport chain through the series of redox reactions, they lose the energy, as we've talked about, because each protein is at a lower energy level. And this energy is used to actively transport H+ ions, protons, across the membrane. So if you look over here, you can see my protein. So this is my electron, one of my electron transport proteins. That is an electron acceptor, but it is also a channel, a protein channel. My H+ ions are being actively transported from the stroma into the thylakoid space. The thylakoid space, remember, is the space inside that little sac, that little bag that is the thylakoid. So, because remember, this is the thylakoid membrane. My H+ ions are being actively transported across the thylakoid membrane from the stroma into the thylakoid space. Why is that important? Well, what that does is it's creating a proton gradient. It's creating a high concentration on one side of the membrane in the thylakoid space. There's going to be lots of H+ ions. And then in the stroma, there's going to be not as many H+ ions. So because we've got this gradient, so we've got lots of H+ ions on one side and not a lot of H+ ions on the other side, we've got a concentration gradient. The hydrogen ions, the protons, are going to want to diffuse down their concentration gradient. And the only way they can do that to get back across membrane down the concentration gradient to get from the high concentration to the low concentration is through a specialized protein channel, which has ATPase, ATP synthase, ATP synthetase. All of these are words you might use to hear it being described, but it is an enzyme that catalyzes the reaction which creates ATP from ADP. So the movement of the H plus ions across this membrane through this protein channel actually causes ATP synthase enzyme to spin, to turn, and that actually causes the catalyzing of the reaction between ADP and ATP. So it phosphorylates ADP, it adds a phosphate group to form ATP. So that driving, because of this chemical gradient that we've created between of H plus ions, is why they call it chemiosmosis. So we're making something using a chemical gradient, a chemical concentration gradient of H plus ions. So as I said, synthase, synthetase, ATPase, different textbooks, different diagrams, different examples will refer to it in different ways, but they all are the same thing. I would try and use synthase or synthetase in the exam, not ATPase, because that is a group that could include, just because it has A's on the end, it could also include enzymes that break apart ATP. So we've got this flow of protons and it's driving that turning, that spinning ATP synthase enzyme, which is helping to catalyze the reaction and phosphorylate ADP to ATP. So because this is driven ultimately by light energy, if we didn't have the light energy, we wouldn't be exciting the electrons, which wouldn't then be able to move, have a higher energy level to be able to move down the electron transport chain and lose energy as they do that, because they wouldn't have had enough to start with if they weren't excited by the light energy in the first place. And if they weren't moving down the electron transport chain, the H plus ions would not be, there wouldn't be any energy to actively transport the H plus ions into the thylakoid space. And if there wasn't a gradient, if we couldn't do that, we wouldn't have the concentration gradient, the difference between low and high either side of the thylakoid membrane. And therefore there wouldn't be a concentration gradient to cause those H plus ions to diffuse through ATP synthase. So this is driven by light. So we are phosphorylating ADP to ATP, and we're using light, so that's why this is known as photophosphorylation. Okay, so this use of electron transport chain and the chemiosmotic theory of the movement of H plus ions is what is creating ATP in photosynthesis. So we call it photophosphorylation. We're going to come to respiration and we call it something different when we talk about respiration, even though it works in a very similar way. Just to note that this happens at all electron transport chains in the thylakoid membrane in photosynthesis, in the light-dependent reactions. So this happens from between photosystem 1 and photosystem 2, this happens. And then we're going to talk about in a minute how it also happens at photosystem 1. So the creation of ATP through this photophosphorylation happens at both photosystems, just in two slightly different ways. So there's a part of the end to the story that we haven't talked about yet, but both are doing photophosphorylation. Okay, so there's a lot of big words in here now, and it depends on your exam board whether they refer to it in this way or not. But this is kind of the full story of the light-dependent reactions and how it gets to the end. We're piecing it all together and how we get to the point where we make a product that goes to the light-independent reactions. So this is referred to as non-cyclic photophosphorylation, and I'll explain why. It could just be referred to as photophosphorylation. Ultimately, it's just how we make the ATP and the reduced NADP. This process uses both photosystem 1 and photosystem 2. It produces reduced NADP, or NADPH plus H+, which you can write it as that as well, and the ATP, and they are both needed for the light-independent reactions. Okay, so that's the point of the light-dependent reactions, is to make things that we then need for the light-independent reactions. And in this example, because we said we're going to talk about phosphorylation in respiration as well, NADP is the final electron acceptor. So it moves through all these various proteins and electron acceptors and photosystems, but it finally ends up in the last place the electrons go is NADP. Okay, so let's start at the beginning, and this is a bit of a recap of what we've already talked about. So we have light hitting photosystem 2. that light energy being used to excite two electrons, which then leave the chlorophyll molecule. They go to a higher energy level and they get accepted by the primary electron acceptor. It leaves the chlorophyll ionized, so it is an ion, and therefore able to get or wanting 2 electrons. Then we have our electron transport chain. So the electron that's left the chlorophyll, gone to the primary electron acceptor, it moves down the electron transport chain through the series of redox reactions, losing energy as it goes. And that energy is used to generate ATP through chemiosmosis, as we've talked about. You normally don't have to talk about the full explanation of chemiosmosis here unless the question is specifically asking you to focus on that part. So be aware that when we discuss photosynthesis, a lot of the time, unless it's specifically asking you to go into detail about how ATP is made, when describing the general pattern and the general kind of way that the light dependent reactions go, we can just at this point that electron energy lost from electrons moving through the electron transport chain is used to make ATP. Then, because our electrons come from photosystem 2, it arrives at photosystem 1, which is at the end of that first electron transport chain, and they've got low energy at this point because they've gone through a lot of electron carriers. So then we've got light coming in again, and then that light energy is going to re-energize, re-excite our 2 electrons, which are going to leave the chlorophyll again at photosystem 1 this time, and they'll go to another primary electron acceptor, and they'll pass along another electron transport chain. This one is shorter, okay? So it doesn't generate as much ATP as the one between photosystem 2 and photosystem 1, but it still is an electron transport chain. It passes through some electrons. And what it will end up going to is that it will end up joining, along with 2 H+ ions, NADP. So it reduces NADP. Remember, reduction is gain. It's gaining 2 electrons and 2 protons. So it's becoming reduced NADP, or NADPH plus H+, which is how you can write it. They will accept different ways of writing it in the exam, but I like to stick with saying reduced NADP because then you're not getting yourself in a pickle over the H pluses and the H's and whether you've got them or not. NADP is technically classed as a coenzyme. It can be referred to as an electron carrier as well, but we've reduced it. So the final electron acceptor, where the electron ends up at the end of that short electron transport chain from photosystem 1, is NADP, and it binds with a couple of H+ ions and it reduces the NADP. And now that NADP is important, the reduced NADP, is important because that's needed for the light independent reactions. We've made some ATP and we've made some reduced NADP. And both of those are now going to be in the stroma and available for the light independent reactions. So this is where the cyclic, non-cyclic part comes in. So obviously this is photophosphorylation. We're using light to phosphorylate, so to add phosphate group to ADP to make ATP, and we're making reduced NADP as a result, because if we didn't have reduced NADP at the end to be the final electron acceptor, then there would be no gradient, there would be no movement of electrons through this whole process, so we wouldn't make any ATP. But the reason it's non-cyclic in this example is because once the electrons have left photosystem 2, once they've been lost, they've left photosystem 2 and gone to the primary electron, etc., they must be replaced somehow, come back. They don't return as a cycle. They are gone. So they have to be replaced by photolysis. And now we said photolysis only happens at photosystem 2 because that's where the enzyme is, and also because that's the only place that electrons need to be replaced. You'll notice photosystem 1 obviously gets electrons from photosystem 2 at the end of that electron transport chain, but there's also another part that makes it slightly different, which makes it cyclic, which we'll talk about next. Okay, so I've sort of edited our diagram a little bit so you can kind of see this other part of photosynthesis in the light-dependent reaction. So this is what's known as the cyclic photophosphorylation. All of these 2 parts, the cyclic, the non-cyclic, are happening at the same time. They're happening concurrently, and it's just a way of describing how the electrons move in 2 different ways. Here, we don't need photolysis to happen. This only involves photosystem 1. It's only talking about how electrons move around from photosystem 1 and back to photosystem 1. This doesn't involve making reduced NADP. This is just about making some ATP from the electron transport chain that comes from photosystem 1. So same as before, we talked about electrons arriving at photosystem 1 from photosystem 2 and that electron transport chain. And we know that that happens. Okay, we've seen that happen here. Photosystem 1 doesn't really need its own electrons because it's getting them from photosystem 1, but sometimes it also gets its electrons itself that come back. The electrons that get excited rise to a higher energy level. They can go through the electron transport chain through a primary acceptor and go on to reduce NADP. That's non-cyclic. Cyclic would be when the electrons this time actually pass through the small electron transport chain, a few proteins in the membrane, and then what they actually do is they return back to the chlorophyll in photosystem 1. So they return and then they get re-energized by light and then they go round again. So you can see how this is a small little cycle. It's cyclical. The electrons are just moving through a short electron transport chain and lowering their energy levels and then coming back to the chlorophyll to be re-excited again by light and then come back down. So that's why this is known as cyclic. It only makes a small amount of ATP because the electron transport chain is short, so there isn't a lot of energy released through here. And it just means that we can keep doing this to keep making ATP. We aren't making any reduced NADP, but we can just keep doing this to make ATP if we are, say, low on electrons coming from photosystem 2. So here you can just see two diagrams just kind of summarizing it. So non-cyclic, we are using photolysis here. We've got our chlorophyll and our light energy. We've got excited electrons, and then they are moving down the electron transport chain, and then that is making energy for the production of ATP. And also it is reducing NADP to NADPH plus H+. Whereas with cyclic, this is, remember, only happening at photosystem 1. We've got our light energy, no photolysis going on, and the electrons are excited, and then they travel down the electron transport chain, and then they come back to the chlorophyll. And that moving through the electron transport chain creates some energy for production of ATP. But in series, that is cyclical. It goes round in a circle. This one doesn't. It ends up going to the electrons go to NADP, and we have to replace those electrons that are being lost. Okay, so this is a summary diagram. So we're going to just go through the stages. If this was a question asking you how to describe the light-dependent reactions that take place in photosynthesis, this is sort of how I would describe it, how I would break down my answer. So light energy hits chlorophyll at photosystem 2, and light energy is used to excite electrons, raising them to a higher energy level. They leave the chlorophyll, and then chlorophyll has been ionized or becomes an ion. The electrons that are then lost from chlorophyll are replaced through photolysis, which is the process of splitting water into oxygen, hydrogen ions, and electrons using light energy. So these electrons from this reaction are used to replace the electrons that have been lost from the chlorophyll molecule in photosystem 2, and it only happens in photosystem 2 because there's an enzyme there that allows it to happen. So the excited electrons that have left chlorophyll then travel to or bind to an electron acceptor, and then they move down the electron transport chain, losing energy as they go through a series of redox reactions or alternating reduction and oxidation reactions through a series of electron carriers or electron acceptors. So we use the two electrons from the photosystem 1 that have moved through some protein carriers or electron carriers, electron acceptors, and also two hydrogen ions as well, which obviously we know are in the stroma because of what's happening with the electron transport chain and the gradient of H+ ions. So some H+ ions and some electrons are used to create reduced NADP. So now here, because we're at our ATP synthase enzyme, we're talking about the fact that the energy lost from the electrons moving through the electron transport chain is used to generate ATP through chemiosmosis, which obviously we've explained before, and you could go on to explain that if you wanted to. Some questions will require it, some won't. And then finally, we can sum up by saying that the ATP and the reduced NADP are then in the stroma and are available to be used for the light-independent reactions. Okay, so final summary then. So we've had, that's the overall general summary that you'd use for, say, a long answer question about the light-dependent reactions. This is just a summary of the products, which is important because we're going to talk about this for this section and the light-independent reactions because you'll see how they link. So oxygen is obviously, oxygen gas is one of our products from these reactions of photosynthesis. So it's made through photolysis, which obviously happens at photosystem 2. And it leaves the chloroplast, it diffuses through the membrane, the inner and outer membrane of the chloroplast. It's going to go into the cytoplasm of the cell, of the plant cell, and then most likely it's going to end up diffusing out of the leaf through the stomata, unless it's used in respiration. Then we have to think about the other product. So we're going to be making, the whole point of these reactions is going to be making ATP, and we need to make ATP for the light independent reactions. And we make it through the photophosphorylation. So whether it's cyclic, non-cyclic, it doesn't matter. It's made at both. We make it through there at both photosystems, and we need that ATP to be used in the light-independent reactions, and we'll see where that gets used in the next video. And then we're talking about our last and final one is reduced NADP. You can say NADPH. You can say NADPH plus H plus. Okay, and that's being produced again from photophosphorylation. It's the final electron acceptor. It's how we get the end of the electron transport chain through both sets of photosystems. And that's also going to go to the stroma for the light independent reactions because we need it for that as well. And that's it. That is your summary of the light dependent reactions of photosynthesis. Hopefully that made sense, and we'll talk through the next part, the light independent reactions, in the next video.
Ouch! This is why in some videos I have unexplained scratches.